Yeah, thank you, Min, Rink, Moon, and everyone else on the organizing committee for inviting me to talk. I'm really happy to share with you guys some of the work that I've done and the thoughts that we've developed over many years of being part of the iCiv community. As Min had said, my name is Brian Dobosh. I'm a research scientist at Emory University. I also am a founder of two companies, one of which builds lab automation equipment like pipetters and automatic tube labelers, which is not relevant to today's talk, and another that is using EVs as therapeutic delivery vehicles to create next generation immunotherapies. And I am the chief technology officer of that, and so I have to disclose that for today, which is the first time I've ever had to do that. So the points that I want to get across today really comes back to some of the, I guess, fundamentals of EVs. Why are they special? We're all here for a particular reason, but EVs and the cargo that they contain behave differently than if that cargo were just on its own. In addition, we heard this morning about a lot of different immune cells that EVs are interacting with, and these immune cells may be a barrier for delivery, but they might also be a good target for delivery. So that's the last point that I want to focus on, is that EVs can be immunotherapies and target particular cell types in addition to particular tissues. When I think of EVs, there's many things that are good about them. They're generally considered very stable, and getting them, particularly from blood, is considered non-invasive, especially compared to other tissue biopsies or invasive techniques like bronchoalveolar lavage to get samples from the lungs. But there's also something else that is less rarely spoken about, and this is called carrier effects of EVs. And this was summarized very well in a paper from Phil Askenaz in the 2021 article of RNA Biology. I highly recommend that people read about it. And he covers 3 points, which are that EVs concentrate the molecules within them, they can protect cargo from changes in pH and degradation by RNases, and they can also target cells for delivery. And I like to add a 4th one, which is that they can alter the biophysical properties of their cargo. And so here are some figures that I'm preparing for a review article. Might be tough for some people to see them, but within the blue box, I have a lot of free-floating molecules around and 2 cell types that have receptors on them. In the red box, I have EVs with the same number of these particles floating around, but now they're concentrated on EVs. And if I had the ability to do a Western blot, ELISA, or some other quantitative assay on these molecule X within the blue box, I would get a particular value. And if I did that same Western blot or other quantitative technique for this molecule in the red box, I would get the exact same number because there are the same number of these particles in both samples. However, if I look within the local environment, what is the cell actually experiencing? In the blue box, this molecule is now diffuse and evenly distributed throughout the environment. Whereas now when it's concentrated on EVs, that increases the local concentration and therefore the avidity of signaling. And now this cell experiences a much greater local concentration of that molecule compared to this one. And so EVs alter the signaling dynamics of whatever cargo is on their surface. As I had alluded to earlier, EVs also protect their cargo from degradation. So in the blue box, I have a generic nucleic acid. Maybe it's a microRNA, maybe it's an mRNA, and that could be very quickly degraded by RNases in circulation. Maybe it's pH in the stomach and they get degraded that way. But conversely, that RNA within EVs is now protected from degradation either by a pH change or inaccessibility of the RNases to get to it. And now if that EV were within circulation or another tissue, it can survive for a little bit longer. The third point that I want to bring up is that EVs also change the potential for delivery. This one's a little bit more complex in that I do have an RNA or small molecule that could potentially bind to a receptor. And in this case, I have 3 of these molecules, and they're evenly distributed amongst these 3 cells. 2 different cell types, one in blue, one that's pink and looks a little bit like a neutrophil. I'll get to that in a little bit. However, now that molecule is packaged within EVs, And those EVs have their own receptors or ligands on them that target them to a particular cell type. And now these generic blue cells that in my review I'm calling monocytes no longer have access to those EVs or are less preferentially targeted by those EVs, which means there's a lower probability that they receive that cargo. In contrast, the neutrophils have a receptor that recognizes some molecule on the surface of that EV, and now the cargo within that EV is preferentially delivered to a neutrophil. And I might not have caught this if I was just looking within a sample. I would have still calculated, again, concentration-wise, this small molecule or cargo within the EV would have been the same in the blue and the red box. Maybe I wasn't looking at cellular delivery in vivo, but now that they're packaged within EVs, this cargo only gets delivered to neutrophils, or at a very high percentage is preferentially delivered to neutrophils over the other cell type. And the last point that I want to bring up is that EVs change the functional properties of what could be on either associated within them or on their surface. EVs have a range of sizes, but if we were to give them a diameter of 100 to 200 nanometers, one molecule of something within their inside of an EV has a concentration of micromolar to millimolar, which is tremendously greater than if that one molecule were within free-floating fluid. And so kinetics that may be unfavorable in a test tube suddenly become very favorable if they're packaged within a particle. This is one enzyme, neutrophil elastase, that we study in our lab, as well as many collaborators. It's not important what it does, but we have many ways to quantify its activity. And there is a natural protein inhibitor of neutrophil elastase called alpha-1 antitrypsin. In the left, we have recombinant free non-EV associated neutrophil elastase that has a particular activity. And if we add the protein-based inhibitor of it, alpha-1 antitrypsin, or a small molecule-based inhibitor of it, the activity goes away, which is what you would expect to see in a test tube. But now, if we take the same concentration of neutrophil elastase that is now associated with EVs, and that's shown in this lighter green line, we have that same activity as we have on the left. If we now have EVs and we incubate them with the alpha-1 antitrypsin, the protein-based inhibitor, we do not have the same flat line on the right graph as we do on the left. Because the elastase is associated with an EV, it is no longer accessible due to steric hindrance by its natural protein-based inhibitor, even though the concentration is exactly the same. The small molecule in this case is still able to target it, just not quite as efficiently as if it was free-floating. Now, maybe this enzyme could be degradative to the tissue around it, as is the case here. Maybe it's a therapeutic enzyme that you've now engineered to be on there. But because it is associated with an EV, it behaves differently than if it was just in a test tube, and this needs to be considered. The other thing is that you may have an enzyme that is a mixture of EV-associated as well as soluble. And those dynamics, the on and off rate of it has to be considered as well. This is a figure we saw earlier, and of course Ida is the original creator of it. There are many types of EVs out there, some of which are present in blood, some of which are only within particular tissues, and they all have different combinations of the earlier carrier effects that I brought up. In addition, there are many, many different immune cells, as well as non-immune cells, that these EVs, or extracellular particles, are going to come into contact with. And in particular, I'm going to talk about the neutrophil, which, as Pia and others had said, makes up maybe 50 to 70% of our circulating white blood cells. And maybe 2,000 to 3,000 red blood cells are present for every one neutrophil. Other subsets are present to a much lesser degree. Monocytes might be 10%, leukocytes, T cells, 20%, and NKs at a much smaller percentage. And the reason I bring up neutrophils, in addition to them being highly prevalent, is that they're also highly penocytic. Neutrophils have a variety of functions. They are the first responders to many stresses that occur within our body. And because they're highly penocytic, they are removing debris. They're not just killing bacteria that may be present. And so because of this, red blood cell EVs and platelet EVs will tend to be preferentially taken up by neutrophils. Now, for many of you who are developing new therapies, that might be a problem. And that's actually one of the reasons why red blood cell-derived microvesicles haven't been very prominent within the delivery space. I think another point for discussion could also be, how do EVs compare to lipid nanoparticles, polymers, viruses? I'm not going to get too much into that in this talk, but happy to talk about that. This is a couple of papers that I've enjoyed reading where they use different methods of generating microvesicles or particles from red blood cell cargo. In the top one, this was a paper that showed that red blood cell EVs can deliver a variety of transcription factors, small and larger RNAs, proteins, as well as small molecules to neutrophils. And those neutrophils had altered functional properties as a result of those deliveries. This is one example where they were looking at NF-kappa B, but they went through a variety of molecular techniques. It's an older paper, so they don't have the array of biophysical characterizations that we would use nowadays. But I do think it's been repeated in many different papers for different subsets of neutrophils and EVs. The second one is an engineering article where they formed microvesicles, labeled them with a dye, and then delivered them at differing ratios to neutrophils. And these CFSE labeled red blood cell EVs were preferentially taken up by neutrophils compared to, say, T cells or monocytes that were also co-incubated with them. Again, this is driving home that EVs don't behave the same way with every cell type that they're coming across. Different cell types behave differently. And that might seem obvious when said out loud, but I do think it's something that's not looked at nearly enough. And in addition to that, it's difficult to look at many different effects of EVs. We spend a lot of time on rigor and standardization here, and that is absolutely great, but it also takes a lot of material to conduct all of these tests. And so now to add in different cell types, different incubation conditions, it does become quite challenging. In terms of engineering EVs to load in custom cargo, there's what I'm sure is going to be a great talk coming right after this. So I don't want to belabor these points, but there are many, many proteins that are preferentially loaded into EVs over others. Highlights are the Lin signal peptide, which is commonly called PALM for palmitoylation motif, tetraspanins like 63, 81, and 9, as well as many others. And there's a paper from 2021 from a company that did a really great job of taking these proteins that are preferentially loaded into EVs and attaching a gene of interest. Here it's a generic GFP, but it could be a luciferase as another reporter or an actual therapeutic cargo. And they showed this as a method of engineering EVs. And I think this is something that still bears a lot of new developments. We haven't done a lot of directed evolution of these protein loading motifs. We also haven't done a lot of optimization nor characterization of, do multiple EVs all have the same amount of cargo, which is almost certainly not going to be the case. Are there subsets of EVs that no longer have these engineered cargoes, and how can we purify them so that we have a more efficacious population when we actually do go into therapeutics? In addition, when we look at an entire EV population and we have a protein that we care about, say for a diagnostic assay, most of the particles around are likely to be noise in that measurement. So how can we specifically isolate the EVs that we care about? Well, I don't have an answer for that. Hopefully some of you do. Something that's not spoken about nearly as much in terms of engineering of EVs is RNA loading into them. There's probably a variety of reasons for that, but I do want to draw attention to a paper from a couple of years ago, which looked at motifs of RNAs that were preferentially loaded into EVs or not. And I think a lot of the techniques that have been, or principles that have been applied to protein-based engineering of EVs can be applied to RNA-based engineering of EVs, such as taking some of the motifs that have been preferentially loaded into EVs by a cell and using that to now engineer custom RNA-loaded EVs. There are challenges such as the cell type matters, the production method, purification techniques, all of the things that we're hearing about today still become relevant. But I do think this is a field that merits a lot more work, especially as RNA detection methods and effects are better studied. As a proof of concept, I have two figures delivering nanoluciferase or GFP to neutrophils or epithelial cells in cell culture. We've done some of this work also in ferrets to show utility of gene transfer. But here we took the Lin signal peptide or the palm motif conjugated to nanoluciferase, and we showed that this can be delivered to human neutrophils and that they actually contain catalytically active luciferase after a couple of hours. And this is fairly basic, but neutrophils, which are considered to be highly proteolytic, they're not considered a target for therapy. This is another one where we had mRNA transfer with a particular motif in their 3' UTR that loads that RNA into EVs. We then took those EVs and delivered those to primary human neutrophils isolated from blood. In addition to having some activity from protein that was also loaded into those EVs, the mRNA was able to be translated. We've done studies with hexokinase as well as ribosome loading to help verify that. But I know I haven't gone through all of those controls for this sake of time. And delivery can also be synergistic. So just like the carrier effects may be present all at the same time, the ability to engineer cargo, you can take a lot of different techniques from different groups and create an optimized EV. They're still optimizing the purification methods and standardizing it, but we don't have to recreate the wheel every single time. We stand on the shoulders of giants. Let's make sure that we all take the best methods of EVs from everyone. On the right, we're showing very similar principles, but we can also add targeting antibodies. So we're trying to deliver to neutrophils, or we're trying to deliver to what are called H441 cells, which are the epithelium, and they're loaded with GFP. So with no targeting, EVs that are co-incubated with both cell types at the same time, go to neutrophils, about 60% or 40% to the epithelium. Again, neutrophils preferentially take up EVs because they are highly pinocytic. But now if we add an antibody that targets them towards the epithelium, this is EpCAM, all of a sudden the epithelium receives about 60% of the EVs and 20% of the neutrophils do. And again, we can multiplex these engineering techniques to say not only are we targeting the epithelium, but we're adding don't eat me signals, which were alluded to earlier. Now again, 80% of epithelium get it and only 20% of the neutrophils do. And we can reverse this also with other antibodies such as targeting CD14, which is on myeloid cells. Again, the point of this is just that their EVs can be engineered in multiple ways, and that the principles of putting RNA or protein into or on EVs or targeting in particular cell types can be therapeutically used, but also is naturally occurring in all of the EVs that we are studying. And so with that, I urge you to think about how the EVs are actually behaving once you have your biomarker of interest. What is the actual mechanism? Is an EV actually special for what you're studying? Or is it the dirtiness of the sample that's relevant? Is it soluble? Is it EV associated? Is it part of the corona or inside? So with that, I want to thank first you guys for listening to me talk, as well as the organizers, as well as an incredible lab that has helped me get to this point.
Thank you, Brian, for the wonderful talk. We have some questions from the chat now. The first question is about EVs carrying virus. If EV can carry virus particles and can potentially cause infection.
Yeah, I think it definitely depends on the context of that. I think it's been well shown by many groups, not ours though, that EVs and AAVs can be associated with one another. And there's a lot of interest in finding the AAVs that are contained within an EV so that they are not targeted by circulating antibodies. There's been work that SARS-CoV-2 and influenza can cloak themselves within EVs, and HIV, part of its mechanism of actually forming its virion is the same thing as the exosome biogenesis pathway. So yeah, definitely EVs and viruses can absolutely co-associate or be the same particle.
Great. We have another question there.
Brian, thank you. Can you hear me? Yeah. Great talk. And I wanted to connect the first part and the last part of your talk a little bit. So you were saying there could be proteins, cytokines or so, binding to the EVs, which would have a much more selective or differential effect on the cells. But for the therapy delivery, wouldn't it be also that if a cytokine would be bound to an EV, it's less likely to be taken up and therefore taken out of the pool? So you will have a longer lasting response because it could detach, attach to the next cell. So you would have a multitude of the responses in several cells instead of one per cytokine. So could you use this for therapy delivery if you actually want to induce a pathway or do a reaction in the cell, that this could be a longer lasting or more effective way of therapy delivery?
Yeah, it brings up an interesting point, and I didn't cover it, but right, an EV doesn't have to actually be taken up by a cell to have an effect, and that's true for, I guess, every molecule in our bodies, potentially. So right, if there's a molecule on the surface of an EV, it interacts with its receptor on whatever cell type, then that EV moves to the next cell, and that effect can now be longer lived. Those kinetics, both the temporal and the spatial ones, have to be considered and could be really cool and interesting to study. Thanks for bringing that up.
Great. We have more question here. Which is more preferable? It seems like this one not relevant. Okay. Can we differentiate between host EVs and other bacterial EVs residing in the GI tract?
Yes and no. It depends on the technique and the standards that are used. In some ways, a bacterial EV and a human host-derived EV, might show a similar size profile, depending upon the techniques that were used to characterize it. But there will also be differences in terms of some of their cargo. So maybe a bacterial EV, we can detect the LPS on it with an antibody, whereas presumably an immune cell or epithelial-derived EV doesn't have that LPS or whatever marker. But maybe also those EVs can become duplets, and they're associated with each other. Now we can't really tell the difference. It all depends on the techniques that we have in our arsenal.
I see. Any question from here? One question here which might be relevant. What is the most effective way to separate RBC-generated EVs?
RBC and what EVs?
The EVs from RBCs from red blood cells.
What was the other cell type that you mentioned?
No, the question is just
RBC-EVs.
about the method of purification.
I think that is a question that is better suited for someone else to answer. We have been doing a mixture of a very gentle centrifugation followed by size exclusion, but I am by no means saying that is the best. It's one that has been working for our use and our application, and that might not be best for someone else.
Yes, maybe Sarah will come up next and talk about it, and we can also talk more about the discussion. Please join me to thank Brian. Thank you.